Hello guys, this is your microbiology sections and in this section we will be talking about the basic microbiology, the clinical microbiology, the mycology, the parasitology, the virology, systemic microbiology, and some antimicrobial drugs. Come and let's begin with the basic microbiology. In this section we will first talk about the bacterial structures. Here is your bacterial structure. This is your gram positive bacteria and this is your gram negative bacteria. There is some similarities between these two mentioned in the center, and there is also some differences between these two mentioned on the sides. So come, let's see the gram positive bacteria. Unique thing about the gram positive bacteria are lipoic acids. This is the lipoic acids which is only present in the gram positive bacteria, and in gram negative bacteria unique thing about are the endotoxins. Endotoxins are lipopolysaccharide and lipooligosaccharides, and there is also some porins in the outer cell membrane within the cell, within the lipid, within the this space is called the periplasmic space. This space contains the beta lactamase enzyme which cleave the beta lactam rings of the penicillins. This is unique about the gram negative bacteria, and there is also some similarities between these two. Both have the flagellums. You can see both have flagellum, both have pilus, both have capsules, both have cells wall which are made up of peptidoglycans, and both have cytoplasmic membrane. Come and let's see each and everything in detail. Flagellums are protein in nature. Chemical composition of flagellum protein in nature and function is motility, while the pelafembria are made up of glycoprotein and its function to adhere to bacteria. Adhere of the bacteria of cell surface. 6 pilus forms during the conjugations. I would like to tell you that how to remember the function of the pella and its nature. So I would tell you that end up of the protein, glucose and sugar. Glucose and your protein, that's made up of glucose and protein. So how to remember that? If you want to adhere with the strong relations, be sugary. This is the sugary part, glucose, and this is the adhere to the bacterial cell surface, and this is the pilus. Another thing, specialized structures are spores only present in the gram positive bacteria. These spores are keratin like coat, diplocholiacids, peptoglycan, and its function is survivals. Resist dehydrations, heat, and chemical. If you want to kill these spores, you need to heat up the medium or the anything which you suspect that these might contain spores. You need to heat it for 121 degrees centigrade for 15 minutes. Then this spore will be denatured. Capsule envelope. Cell envelope are capsule's chemical structure is directly or discrete layer usually made up of polysaccharides and rarely protein in nature, and this capsule protect against phagocytosis. And slime layer, S layers, is loose network of polysaccharides This S layer's function is mediate adherence to the surface, play a role in biofilm formations like end-dwelling catheter. This S layer is present in bacteria step epidermidis. Outer membranes are outer leaflet. Outer leaflet contain endotoxins. This outer membranes is only present in the bacteria. Bacteria have outer membranes. Within the outer membranes, there is embedded protein, and there is also inner membranes or inner leaflet. Outer leaflet or outer membrane contain endotoxin, lipopolysaccharide, and lipooligosaccharides, while embedded proteins are porins and other outer membrane protein OMP outer membrane protein. Inner leaflets are made up of phospholipids. Phospholipid bilayers. This present only in gram-negative bacteria, while endotoxin lipid A induce tumor necrosis factor and interleukin 1 antigens or polysaccharide components. Most outer membrane proteins are antigenics, while porine transport across outer membranes. Periplasm. Periplasm is the space between cytoplasmic membrane and outer membranes in gram-negative bacteria, only in gram-negative bacteria. Okay. Peptidoglycan is in the middle. Function of this accumulate component exiting gram-negative cells, including hydrolytic enzyme, example beta-lactamase. Cell wall. Cell walls are peptidoglycans is a sugar backbones with peptide side chains cross-linked by transpeptidases. Net-like structures give rigid support, protect against osmotic pressures and damage. These are the functions of cell wall. Cytoplasmic membrane. Cytoplasmic membrane are phospholipid bilayer 6 with embedded proteins. Example penicillin binding proteins and other enzyme. Penicillin binding protein to which the penicillins bind to these. Lipoticoic acids are gram positives and only extend from the cell membrane to the exterior. This is the lipoticoic lipo, and this is the positive. This is present in gram positive bacteria. The function of cytoplasmic membrane, site of oxidative and transport enzyme, penicillin binding proteins involved in cell wall synthesis, and lipoticoic acids induce tumor necrosis factor and interleukin 1. Come here and see some staining technique by which we can study these organisms. One of them is the gram staining technique. In gram staining technique, this is the first lab technique. Our first live lab technique and bacterial identifications. Bacteria with thick peptidoglycan layers retain crystal violet dye. Gram positive bacteria with thin peptidoglycan layer turn red or pink like gram negative bacteria with counter strains. These bugs do not gram stain wells. These lethal. How to remember which drugs do not stain? This is the formula. These lethal microbes may unfortunately lack real colors but are everywhere. Okay, T for treponemia, L for leptosporia, M for mycobacteria, M for mycoplasma, U for uriplasma, L for legionella, R for rickettsia, chlamydia, bartonella, anaplasma, Ehrlichia. Sorry, I have a little bit throat infection, so due to that reason I cough again and again. So for this disturbance, I am really sorry. So treponemia, leptospiria, To tend to visualize, mycobacteria are cell wall has high lipid proteins, and mycoplasma ureaplasma has no cell walls, and the Legionella, Rickettsia, Chlamydia, Brettonella, and plasma. All these primarily intracellular. These microbes are primarily intracellulars. Also chlamydia, classic peptidoglycan because of decreased muscarinic acid, decreased muramic acid. Gemza stains. Gemza stain is another technique for staining the bacterial microbes. It is used to study the Chlamydia, Rickettsia, Treponema, Borrelia, Helicobacter pylori, and Plasmodium. Come and let me show you the Treponema. This is your Treponema species, okay? And this is the Treponema species. Treponema, and how to remember? Gemza stains, that these microbes stain with Gemza. So Chlamydia, clumsy, Rickettsia is rick, and Treponema is tripeds, and Borrelia is borrowed, and Helicobacter is helicopter, and Plasmids is plastered and gem. So clumsy, ricky, tripeds, and borrowed, helicopter, plastered, and gems. Another technique is PAS stains, periodic acid scap stains. This stain glycogens, mucopolysaccharide used to diagnose the Whipple disease, Treponema whippelis. This how to remember? Periodic PAS stains these microbes as pass the sugar. So come and let me show you the whippeli. Here you can see the whippeli there as the whippeli there, and this is also the whippeli. This is also the whippeli. This is the periodic acid scap stains. ZN stains, Z. Nelson stains, carbol fissions, acid fast bacteria like mycobacteriums, nocardia stains mycolic acid in the cells, protozoa like cryptosporidium oocytes. Ramine rhodamine stains is more often used for screening, inexpensive due to it is because this is used most often because of this is inexpensive and more sensitive. Come and let me show you the Mycobacterium tuberculosis. Here you can see the Mycobacterium species. This is the Mycobacterium species. This is another one. This is another one. This is another one. And this is another one. And this is the another one. This staining is ZN staining. ZN staining. Another staining technique is India-Eng staining. India-Eng stainings are stains the following organism: Cryptococcus neoformans, Mesicaramines can also be used to stain thick polysaccharide capsules red. And let me show you the Cryptococcus neoformans in India, and this is the Cryptococcus neoformans. Hollow structures within inside. This is the Cryptococcus neoformans. India ink. Another staining technique is silver staining. Silver staining technique is used to stain the H. pylori, Legionella, Brettonella, Encella, and fungi. And fungi examples is coccidiosis. Coccidiosis. Here you can see the coccidiosis. This is 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 also the coccidiosis. So this is the silver staining techniques. It's also used to stain the Pneumocystis jirovecii aspergillus fumigatus. How to remember silver stains? Stain these techniques. Helicopters are silver. Helicopters are silver. Helicopter, Helicobacter pylori, Alpha legionella, coccidiosis, pneumoconiosis, aspergillosis. These are silver staining. Fluorescent antibody stain. This is another technique. Fluorescence antibody stain is used to identify many bacteria, viruses, Pneumocystis jirovecii, Giardia, and Cryptosporidiums. Example are as FDA, ABS for syphilis. Special culture. There is some bacteria which do not stains in a particular medium, so these required some special culture techniques. Are there some bacteria which you can also study with some special culture requirement? Following are the bugs which required the special cultures. Number one is H influenza. H influenza is media required or media used for isolation is chocolate agars, and this media contains so the media contents for chocolate agar is factor 5 NAD plus and factor 10 hematenes. Other bugs is Neisseria gonorrhea, Neisseria meningitis. The media required for this is the Thayer-Martin agars, and the media contains selectively favor growth of Neisseria by inhibiting other growth of gram-positive organism, like with the help of vancomycins, gram-negative organisms except Neisseria, and Neisseria with trimethoprim's and colistines, and fungi with nystatins. This media contains also some antibiotic to prevent the other bacterial growth. Like for example, vancomycin is used to prevent the gram-positive organism and gram-negative organism except Neisseria, while in fungals we add the nystatin to prevent the fungal growth. Vancomycins, trimethoprim, and nystatins, these contain the this mediums. Very tightly culture Neisseria. Okay, very tightly cultures Neisseria. This is how to remember this. V for vancomycin, T for trimethoprims, and N for nystatins. Other microorganism, B. pertussis. The media required for this bacterial growth is borted gingo agars, borteds for Bordotella. You see here is the B. Bordotella, borted, here is the borted, and this is the Bordotella, borted. Both have borted, and you can see that borted used for Bordotella or Bordotella can be growth in the borted gingo agar medium, and regains low medium. This is the regain low medium. This contains is the potato extract, charcoals, blood, and also contain antibiotic. Another bacteria is Corynebacterium diphtheria. The media required for this is Telluritate agars and Loeffler mediums. Another bacteria, Mycobacterium tuberculosis. Mycobacterium tuberculosis, the media required for this is the Lowenstein Jason mediums. metal, brooks, mediums, and rapid automated broth cultures. Mycobacterium pneumoniae. Mycobacterium pneumoniae is Eaton's agars required. This medium required cholesterols, while another bacteria lactose fermenting enterics. Lactose fermented enterics. McConkey agar used for this bacterial staining and fermentation produce acid, causing colonies to turn pink. Another bug is E. coli. E. coli eosin methylene blue. E for E. coli. E for E. coli. E for eosin methylene blue. EMB agars. And this colonies with green metallic sheens. Colonies with green metallic sheens. Another bacteria or another bugs is Brucella, Francisella, Legionella, and Pasteurella. Growth mediums for these microbes are charcoal yeast extract, agar buffered with cysteines and irons. The ELA siblings, Brucei, the ELA siblings, ELA, Brucella, ELA. Here you can see the ELA. ELA siblings are Brucei, Francis, and Legionaris. Brucei, Francis, and Legionaris. And Pastors build the cysteines chapels out of the charcoal and iron. Charcoals and iron. Fungi suborod agar subs are fungi. Subs or rod agar used for fungi growth. This is how to remember. Subs are funny guy. These are the technique for bacterial growth. Special culture requirement, we study all these things. Okay, come let's see some another more thing or study some more about the microorganism, the bacteria. Anaerobes are those bacteria which do not require oxygen for their growth. Example are Clostridiums, Bacteroides, Fusobacteriums, Actinomyces israeli. These lack catalase and are superoxide dismutase and are thus susceptible to oxidative damage. Have you ever seen or you will study these things in the immunology section and also in the biochemistry? There's oxidative enzyme or oxidative chemicals which kills the bacteria. One of them is hydrogen peroxide. Hydrogen peroxides combine with the chloride to form hypochlorous acid, and this hypochlorous acid will kill the bacteria. Some bacteria have catalase oxidase enzyme which convert the hydrogen peroxide into water and oxygen, and these bacteria will be protected from oxidative damage. And anaerobes bacteria don't have these enzymes, so due to this reasons these bacteria are prone to damage by the oxidative radical or oxidative chemical. Generally, foul smelling short chain fatty acids are difficult to culture. These bacteria are difficult to culture and produce gas in tissues like carbon dioxide and hydrogens. How to remember? Anaerobes cannot breathe fresh air. C for Clostridiums, B for Bacteroides, A for Fusobacteriums, and A for Actinomyces israeli. Anaerobes are normal microbiota and GI tract, typically pathogen elsewhere. Aminoglycosides are ineffective against anaerobes because these antibiotics require oxygen to enter into bacterial cells. Facultative anaerobes, these bacteria may use oxygen as a terminal electron acceptors to generate ATP but can also use fermentations and other oxygen independent pathways. Examples are streptococci, staphylococci, and gram-negative bacteria. Come and let's see intracellular bacteria, those bacteria which stay within your cells. These are obligate intracellulars. Examples is Rickettsia, Chlamydia, Coxiella, rely on host ATP. It's host ATP dependent. Stay within the cells when it is really chilly and cool. How to remember? Stay inside the cells. Stay inside when it is really chilly and cool. R for Rickettsia, CH for Chlamydia, and CO for Coxiella. Other intracellular bugs are facultative intracellular. Number. This is the obligative, this is the facultative intracellular. These are the Salmonella, Salmonella typhi, Neisseria gonorrhea, Borrelia burgdorferi, Mycobacterium tuberculosis, Listeria monocytogenes, Francisella, Legionella, and Yersinia pestis. Some nasty bugs may live facultatively. How to remember? S for Salmonella, N for Neisseria, B for Brucellosis, M for Mycoplasma, L for Listeria, F for Francisella, L for Legionella, and L for Y for Yersinia. Other bacteria are encapsulated bacteria. These bacteria are contain in outer capsule structure, so that's why these bacteria are called encapsulated bacteria. Examples are Pseudomembranous aeruginosa, Streptococcus pneumoniae, as shown here, and Haemophilus influenza type B, Neisseria meningitis, E. coli, Salmonella, Klebsiella pneumoniae, and group beta Streptococci. These capsules serve as an anti-phagocytic virulence factor. Capsule polysaccharide plus protein conjugates serve as an antigens in vaccines. These capsule polysaccharide and the protein conjugators can be used for preparation of vaccines. Please shine my case. How to remember this? P for Plasmodium, S for Streptococcus pneumoniae, H for influenza, H for Haemophilus, I for influenza, N for Neisseria, E for E. coli, S for Salmonella, K for Klebsiella, and S for beta Streptococci. These are opsonized and then cleaned by spleen. First, these bacteria get opsonized with the help of complement and antibody systems, and then these are cleaned by spleen. If the patient don't have spleen, a splenic patient, no spleen here. How to remember those bacteria? No spleen here. Have decreased opsonizations ability and thus increase risk of severe infection. You need vaccination to protect against these microbe. If you don't have spleen, no spleen here. Neisseria meningitis, strep pneumonia, and H influenza. In these cases, you need to do vaccination if you don't have spleen. Other things are encapsulated bacterial vaccine. These were the encapsulated bacteria. This is the encapsulated bacterial vaccine. Some vaccine contain polysaccharide capsule. Antigens are conjugated to a carrier protein, enhance immunogenicity and protecting promoting T cells activation and subsequent class switchings. Polysaccharides against alone cannot be presented to T cells. Pneumococcal vaccines, example is PCV13 pneumococcal conjugate vaccines, PPSV23 pneumococcals polysaccharide vaccine with no conjugated proteins. And another thing, H influenza type B conjugate vaccine and meningococcal vaccine is also conjugate vaccine. Some other bacteria, urease positive organism. Urease positive organism are Proteus, Cryptococcus, Haemophilus influenza, Uriplasma, Nocardia, Klebsiella, Staph epidermidis, Staph saprophyticus. These bacteria urease hydrolyze urea to release ammonia and carbon dioxide, which lead to increased breath test, predisposed to storage magnesium aminophosphate stones, in particularly Proteus. P chunks, how to remember these P chunks? Urease positive, you can also use urease positive for H. pylori test, is called urea breath test. How to remember? Urease positive bacteria are P chunks, Proteus, Cryptococcus, H. influenza, Uriplasma, Nocardia, Klebsiella, and S for Staph epidermidis, and also S for Staph saprophyticus. Catalase positive organism. Catalase positive organism. I already told you, and it's before that catalase. Some bacteria have enzyme called catalase enzyme. These catalase degrade hydrogen peroxide into water and bubble of oxygens. It will appear like this, bubble of oxygens, and before it can be converted to microbicidal products by the enzyme myeloperoxidase, like H2O2, hydrogen peroxide combined with chloride, and this chloride will kill the bacteria. With the help of this, the conversion of this is myeloperoxidase, with the help of myeloperoxidase enzyme. Hydrogen peroxide and chloride combine with each other with the help of myeloperoxidase enzyme. Some people have deficiency of this enzyme. People with chronic rheumatic disease like NADPH oxidase deficiency have recurrent infection with certain catalase positive organism. Big catalase positive organisms include Bordetella pertussis, Helicobacter pylori, Burkholderia, Cepecia, Nocardia, Pseudomonas, Legion, Listeria, Aspergillus, Candida, E. coli, Serratia, and Staphylococci. Cat have been to place. How to remember catalase positive organism. Pigment producing bacteria. Pigment producing bacteria. Actinomyces israeli, yellow sulfur granules, which are composed of filaments of bacteria. Israel has yellow cells. Staph aureus is golden yellow pigments. Aureus latens, and aureus mean gold. Staph aureus, golden yellow pigment. Actinomyces israeli. You see Israel yellow colors, and Israel has sand yellow color, or Israel has yellow color sand. Pseudomonas aeruginosa has blue-green pigments, pyocyanins and pyoverdines. Rogillas is green. Serratia marcinis, red in pigments. Think red sriracha, hot sauce. In vivo biofilament production bacteria, in vivo biofilament producing bacteria are stiff epiderm. In vivo biofilament producing bacteria are stiff epidermidis. These bacteria will affect or infect the catheter, the prosthetic device, infections. Another thing, viridians, streptococci, strep mutans, strep sanguinus is form dental plaques and also cause infective endocarditis. Pseudomonas aeruginosa also cause respiratory tree colonization in patient with cystic fibrosis. Ventilator associated pneumonia, contact lens associated keratitis, non-typables uncapsulated H influenza are causing otitis media. Spore forming bacteria. Spore forming bacteria, some gram-positive bacteria can form spore as shown here. This is the spores of that bacteria. This is also the spore. This is the spore. When nutrients are limited, when they will form spores, when the nutrients are limited. Spore lack metabolic activities and are highly resistant to heat and chemicals. Core containing diplocholinic acids responsible for heat resistance must autoclave to kill spores. It is done for surgical equipment by steaming at 21 degrees centigrade for 15 minutes. Hydrogen peroxides and iodine based agents are also sporicidals. If you want to increase the chance to kill these bacteria, you can also add the hydrogen peroxide and iodine base because they can kill the spore. Examples of spore forming bacteria are B. anthracis, anthrax, B. serous food poisoning, Clostridium botulinum toxin, Clostridium difficile, pseudomembranous colitis, Clostridium perfringens, also called gas gangrene, and Clostridium tetani cause tetanus. Bacterial virulence factors. Bacterial virulence factor, these promotes evasions of host immune response. These promotes evasion of host immune response. These virulence factors will try to escape your immune response. Number one, these are capsule polysaccharide, protein A, IgA protease, M proteins. Capsule polysaccharides are highly charged hydrophilic structures, act as a barrier for phagocytosis and complement mediate lysis. Major determinant of virulence. Protein A bind to Fc region of IgG immunoglobulin G Fc region. This protein A will bind your immunoglobulin G Fc portions. By blocking the Fc portion, it will prevent opsonizations and will be unable to phagocytose. This A protein is expressed by Staph aureus. IgA protease, enzyme that cleaves immunoglobulin A. Allow this immunoglobulin A is present in your secretion like your saliva, your tears, your mucosa, your GI tract. And by cleavaging of IgA, bacteria allow to adhere to and colonize mucous membranes secreted by Staph, pneumonia, H influenza type B, and Neisseria. Shines. How to remember? Shines. S for strep pneumonia, H for influenza, and N for Neisseria. M protein help prevent phagocytosis. M proteins is also help prevent phagocytosis expressed by group A streptococci sequence homology with a human cardiac myoglobin molecular mimicry possible underlies the autoimmune response seen in the rheumatic fever. The function of M protein is its mimicry. It's show himself like they are the not the pathogen for outside environment, and there is some similarities between M protein and your own body tissues or proteins, and by doing this the your immune system is not going to recognize this, but under circumstances your immune system goes and react to them, and sometime also goes in the same way they get activated your immune system and attacked on your own body tissues, is like it is happening in the rheumatic fever. Come and let's see some bacterial genetics. Bacterial genetics transformations. In transformation, what doing? Naked DNA, like here, naked DNA inserted into recipient cells, and this naked DNA degrade into uncombined DNA. These uncombined DNA, and these DNAs get incorporated into recipient DNA. So here is, you can see this is part is the recipient DNA, this part the which look the blue is the donor DNA. This cells is called the recipient cell, as this cell is called the transformed cells. Depending upon the genetic materials, this DNA, this bacteria can be fully different than this bacteria. So come in, let's see the theory. In transformation, component bacteria can bind and import short pieces of environmental naked DNA chromosomals, naked bacterial chromosomal DNA from bacterial cell lysis. The transfer and expression of newly transferred gene is called transformations. Features of many bacteria, especially strep pneumonia, H influenza type B, and Neisseria shins. How to remember these? This is the features of too many bacteria, including of them is shins. S for strep pneumonia, H for influenza, and N for Neisseria. Adding deoxyribonuclease degrade naked DNA, preventing transformations. Another thing is conjugations. Here is the cells. You can see this is the, let's suppose this is the F positive cells. F positive cells, this bacteria have plasmid. This is the plasmid DNA. F plasmid contain genes for 6' and conjugation. This F plasmid bacteria, this is F plasmid DNA contain genes which is used for 6' formations here, and for conjugation. This is the conjugation. What going ahead, F positive cells have the plasmid bacteria, while F negative cell don't have plasmid. F negative cells don't have plasmid DNA. Next, F positive cells form a 6' and conjugal binding occurs. This is also called mating bridge. What happens? The plasmid transfer half of its chromosomal materials into the F negative cells. Single strands of the plasmid DNA transfers into F negative cells. F negative cells, and after conjugations, this F negative cells also convert into F positive cells. But remember, there is no transformations of chromosomal DNA. Chromosomal DNA, this is the chromosomal DNA. Chromosomal DNA is not going to be transferred in this process. So F positive cells, F negative cell convert into F positive cell by this process, can be represented by F positive cross F negative. F positive plasmid genes required for 6 plus and conjugations. Bacteria without plasmid, our term is F negative, like this, while 6 plus on F positive bacterium contains F negative bacterium single strand plasmid is transferred across conjugal bridge. This is also called mating bridge. No transfers of chromosomal DNA occur in this process, like I already told you. HFR and F negative plasmid conjugations. HFR mean high frequency recombination. HFR, high frequency recombinations. What happen in this technique? In this technique, the plasmid DNA incorporate into its own chromosomal DNA. High frequency recombinations. Cells contain F positive plasmid incorporated into bacterial DNA. This plasmid incorporated into bacterial DNA, this is the F negative cells. And this F positive cell convert into high frequency recombinations cells. After that, the leading protein of plasmid transfer along with flanking bacterial chromosomes. In this process, the bacterial chromosome with plasmid DNA transfers into F negative cells, and in this F negative cells, the highly high frequency recombinant cells stay the same as like the plasmid within the chromosomes, and this recombinant cells forms by the inserting of these genes into F negative cells. So what's happened? F negative cells bacterial DNA plus F positive cells bacterial DNA plus plasmid copy. This includes the F negative cells, F positive cells bacterial DNA, and also contain the plasmid copy of the DNA. So what happens in this case? F positive plasmid can become incorporated into bacterial chromosomal DNA. It's like I show you highly high frequency recombinations cells. This is the high frequency recombination cell. Transfers of leading parts of the plasmid and a few flanking chromosomal genes. High frequency recombination may integrate some of these bacterial genes. Recipient cells remain F negative, but no may have new bacterial genetics like this. Transductions. Transductions are generalized transductions, specialized transductions. Yes, generalized, here is specialized. What happen in generalized transductions? Here you can see in generalized transductions, the lytic phage or the lytic prophage incorporates its own DNA into bacteria, and this bacteria cleavage of bacterial DNA. When this get enter bacterial DNA cleaved, and bacterial DNA packed in the phage capsid. Bacterial DNA also cleaved, and this bacterial DNA can also packed in the bacterial picked this bacterial DNA picked in the phage capsids. When this phage and capsids, the lysis of the bacterial cells occur, release of newly phage from the lyse cells, and this lytic cells in this phage again affected another bacteria, and by affecting or infecting another bacteria, these genetic materials get transferred into new bacteria. So these genetic material will be different from this ones. This is called the generalized transductions. This is a picking error. Lytic phage infect bacterium, leading to cleavage of the bacterial DNA. Part of bacterial chromosomal DNA may become picked into phage capsid. Phage infect another bacteriums, like it's shown here, another bacteriums, and transfer these genes to this bacterium. This is the generalized. And what happen in specialized specialized transduction and specialized transductions? Lysogenic phage, viral DNA incorporated into bacterials, and in this bacterial, the viral DNA incorporate into bacterial DNA. You can see here they lyses this DNA, but in this case, the lytic phage incorporates its own DNA into bacterial DNA, and the phage particles carry bacterial DNA. Here, lysis of new phage occurs, and when this infect other bacteria, the gene transfer from donor to recipient. So what will happen in this case? You will find the bacterial its own DNA, the phage DNA, and the another bacteria DNA. This is called the specialized transductions. This is an excision event, and this was the picking error. In excision event, lysogenic phage infect bacterium viral DNA incorporate into bacterial chromosome. When phage DNA is excised, flanking bacterial genes may be excised with it. DNA is picked into phage capsid and can infect another bacterium. Gene for the following 5 bacterial toxins are encoded in the lysogenic phage. These are the A, B, C, D's. Group A, Streptococcus, erythrogenic toxin, botulinum toxin, cholera toxin, diphtheria toxin, and Shiga toxin. Another thing is transformations. In transformation, this is the plasmid, this is the transposition genes, this is the bacterial DNA, and this is the target side. What happened? The transformation genes get incorporated into the bacterial genes. This DNA will be different from this one. So what happens? Jumping. This is a jumping process. Okay, jumping process involving transformations. Specialized segment of DNA which can copy and excise itself and then insert into same DNA molecule or as unrelated DNA. Example: plasmid, chromosomes. Critical in creating plasmid with multiple drug resistance and transfer across species lines. Example is TN11546 with 1A from Enterococcus to Staph aureus. Come in, let's talk about some exotoxin and endotoxin. First, let's see within the diagram. This is the bacteria, this is the bacteria, this is your host cells. Exotoxins are within the cells, and when they release, they activate the host cells and downstream cellular reactions occur, while enterotoxins are located within the Membranes, they can be either released or they can be either released by cell itself or can also release during cell lysis. Like for example, we use sometime antibiotic and due to this cell lysis will occur. What happen in this case? The exotoxin get released and will activate the host cell, and the host cell response will be tumor necrosis factor, interleukin 1 and interleukin 6. And exotoxin, there we know interleukin 1, interleukin 6 and tumor necrosis release. There will be some other downstream response. Come and let's see the difference between these two. Exotoxins and endotoxin. Exotoxin source is certain species of gram-positive and gram-negative bacteria, while endotoxin outer cell membrane of most gram-negative bacteria. Secreted from cells, endotoxin are secreted within inside the cell, while exotoxin secreted within inside, while endotoxin have no secreted from the cells. Chemistries. Chemical composition of exotoxin is polypeptides, while the endotoxin chemical composition lipid A component, lipopolysaccharides, structural part of bacteria released when lysis occur. Location of genes for exotoxin plasmid and bacteriophage, while the endotoxins are bacterial chromosome. And toxicity, which one is more toxic? The exotoxin is a highly toxic and fatal dose up to just 1 microgram, while endotoxin are low toxicity, fetal dose an order of 100 of micrograms. Clinical effect of the exotoxin is various effect. We will study in the next page. And exotoxin, endotoxin clinical effect will be fever, shock like hypotension, and DIC, disseminated intravascular coagulations. Mood affections, various mood for exotoxins we will see. In mood affections for endotoxin, induced tumor necrosis factor, interleukin 1 and interleukin 6. Antigenicity. Antigenicity for exotoxin are induce high titer antibodies called antitoxins, while the exotoxins are poorly antigenic. Vaccines are toxoid used as vaccine, no toxoid and endotoxins, and no vaccination available. Heat stability. Exotoxin destroyed rapidly at 60 degrees centigrade except Staphylococcus, enterotoxin, and E. coli heat stable toxin, while enterotoxin are stable up to 100 degrees centigrade for 1 hour. While typical disease occur in the exotoxin are tetanus, botulinum, diphtheria, and cholera. And in case of endotoxin, meningitis occur, sepsis by gram-negative rod. Come in, let's see in detail the bacteria exotoxin. Exotoxins, bacteria, the bacteria is toxin at mechanism of actions and manifestations. Inhibit protein synthesis. Corynebacterium, diphtheria, toxins is diphtheria toxins, and inactivate elongation factor EF2, and it causes pharyngitis with pseudomembrane in throat, and SOR is a lymphadenopathy, bull neck. It can also cause myocarditis, while in Pseudomonas aeruginosa, and exotoxins are exotoxin A. This also inactivate elongation factor 2, and manifestation is host cell death occur. in this case. Shigella species toxins are Shiga toxins, and Enterococcus hemorrhagicus and enterohemorrhagic E. coli. These are both of these species, Shiga and enterohemorrhagic, both of these have Shiga toxin, and both of these inactivates ribosome 60 subunit by removing adenines from ribosomal RNA. Manifestation is damage mucosa, destroy damage GI mucosa, which lead to dysentery and enhance cytokine release. Cytokine release lead to hemolytic uremic syndrome, typically an EHEC. Enterohemorrhagic E. coli serotype O157H7. Unlike Shigella, enterohemorrhagic E. coli does not invade host cell. Another thing, the bacteria which increase the fluid secretion. Enterotoxigenic E. coli, toxin for that heat labile toxin, LT, labile toxin, and another type of toxin, stabile toxin, heat labile toxin and heat stabile toxins. In heat labile toxin or activate adenylate cyclase, mechanism of action, or activate adenylate cyclase increase cyclic AMP, increase chloride secretion in the gut, and chloride is salt, and salt drag water toward itself, chloride and water efflux. And in case of what happened in this case, you will find the watery diarrhea, labile in ear, labile in ear. Labile for watery diarrhea is labile in ear, while stabile on the ground. Here you can see the stable protein, what the stable protein are doing, or activate glutamate cyclase, increase cyclic GMP, decrease reabsorption of sodium chloride, and increase water in the gut. It will cause watery diarrhea. Labile, how to remember which one activate cyclic AMP, which one activate cyclic GMP. Labile in ear, labile in ear, while stables on the ground, they show here. Labile in ear, adenylate cyclase, while stables on the ground is guanylyl cyclase. Bacteria that increase cyclic GMP include bacteria that increase cyclic AMP include cholera, anthrax, pertussis, E. coli. Increase cyclic AMP with CAP. Increase cyclic AMP with CAPE, CAP. C for cholera, A for anthrax, P for pertussis, E for E. coli. So this is how to remember enterotoxigenic E. coli. Increase fluid secretions. Other bacteria is Bacillus anthracis, anthrax toxins. Anthrax toxins secreted by Bacillus anthracis, and mechanism of action mimics adenylate cyclase, increase cyclic AMP, likely responsible for characteristic edematous border of black scars in cutaneous anthrax. Fibro-cholera secrete cholera toxins or activate adenylate cyclase, increase cyclic AMP by permanently activating G-stimulatory proteins or G-stimulatory receptors. Fibrocholeria, voluminous rice watery diarrhea will be seen in this case. Another thing is inhibit phagocytotic ability are the Brordotella pertussis, which secrete pertussis toxins. This activate adenylate cyclase, increase cyclic AMP by inactivating the inhibitory subunit G inhibitory receptors. This will cause the whooping cough. The child cough on expiration and whoops on inspiration. Can cause 100-day cough in adult associated with postosiomesis. Inhibiting release of neurotransmitters. These are the Clostridium tetani and Clostridium botulinum. Clostridium tetani release tetanospasmin, and Clostridium botulinum release botulinum toxin. This also called the tetanus toxin. Both are protease and the SNAR solubles NSF attachment protein receptors, a set of protein required for neurotransmitter release via vascular fusion. Clostridium tetani toxin prevent release of inhibitory GABA and glycine neurotransmitter for Renshaw cells in the spinal cord, lead to spastic paralysis, and rhesus sardonicus, trismus, lockjaw, and opisthotonus are the clinical presentations for Clostridium tetani, while Clostridium botulinum infant botulism caused by ingestions of spore. Examples from soils, raw honey, toxin produced in vivo, foodborne botulinums cause ingestions of preformed toxins from canned foods. And there is another toxin bacteria, lysis cell membrane, Clostridium perfringens. Toxin is alpha toxin and mechanism of phospholipase lecithinase and degrade tissues and cell membranes, and its manifestation as degradations of phospholipids, myonecrosis, which lead to gas gangrenes, and hemolysis. Hemolysis is double zones of hemolysis of blood agars. Streptococcus pyogenes is streptolysine O toxins, and mechanism of this toxin action is protein that degrades cell membranes. This is RBC contribute for beta hemolysis. Host antibody against toxin is all used to diagnose rheumatic fever. Do not confuse with immune complex of post-streptococcal glomerulonephritis. Another thing is Staphylococcus aureus and Streptococcus pyogenes. Staphylococcus, these two are super antigenic, which causing shock. Staphylococcus aureus release toxic shock syndrome toxin TSST1s, while Streptococcus pyogenes secrete and erythrogenic exotoxin A. These toxin cross-link beta regions of the T cell receptors of MHC class II, major histocompatibility complex class II, and antigen presenting cells of the outer antigen binding sites, which lead to overwhelming release of interleukin 1, 2, INF gamma, and tumor necrosis factor alpha, which lead to shock. So clinical presentation of Staphylococcus aureus is toxic shock syndrome, which is fever, rash, shock, other toxin causes scalded skin syndrome, exfoliating toxin, and food poisoning, heat stables, enterotoxins. While Staphylococcus pyogenes is toxic shock like syndrome, fever, rash, shock, and scarlet fever. Exotoxin. Exotoxins are lipopolysaccharide found on the outer membrane of gram-negative bacteria, both cocci and rod, composed of O antigens plus core polysaccharide plus lipid A. The toxic compound Neisseria have lipo-oligosaccharide release upon cell lysis or have living cells by blebs detachment from the outer surface membrane with exotoxin which is actively secreted. Their main effect is affects macrophage activations, T cell, TLR4, tool-like receptor 4 and CD14 complement activations and tissues factor activations. Endotoxins. E for edema, N for, as the name endotoxin represent the effect of endotoxin. Edema, E for edema, N for nitric oxide, D for DIC and death. Disseminated intravascular coagulation. O for outer membranes, T for tumor necrosis factor alpha, O for O antigens, core polysaccharides and lipid A. X for extremely heat stables, I for interleukin 1 and 6, N for neutrophil chemotaxis and S for shocks. This is the endotoxin lipid A components lead to macrophage activations with the help of T tool-like receptor 4 and CD14s receptor activations. By activation these receptors, it will lead to release of interleukin 1 and 6 which lead to fevers, tumor necrosis factors which lead to fever and hypotension, and nitric oxide which lead to hypotension. Exotoxin can also lead to complement activations like C3 and C5A. C3A help in histamine release and hypotension and edema, while C5A also histamine release, hypotension and edema, and also causes neutrophil chemotaxis. Endotoxin also activate tissues factor and tissues factor cause coagulation cascade which lead to disseminated intravascular coagulation.